All right, you guys, let's get through this last lecture of the semester. We are going to cover the reproductive system. So today we're going to be just doing an overview of the reproductive system, and then we're going to go through each male and female reproductive systems separately, see kind of what maybe some similarities are, what are some differences, and then we're going to talk about the process of fertilization. And then at the end, a little bit about the disorders and development. So, what are some general functions of the reproductive system? Well, the whole point is to produce offspring, right? So, each male and female gametes have to come together and produce a fetus, right? So, what is the whole point of that, right? So, each gamete has half your genetic material. You're going to combine those genetic materials together to create an offspring, and that will promote your own genes into future generations. So what are these primary sex organs that are producing the gametes? So when we say gametes, we're talking about sperm in males and eggs in females. So the testes are what are going to be producing the sperm in the male, and ovaries are what are going to be producing the eggs in the females. So we do have accessory sex organs, and these are really to help promote fertilization. That's their whole job. So primary sex organs are producing the gametes, which are the most important things, and then the accessory sex organs are going to promote fertilization. So those are going to be the glands, the external genitalia, things like that, okay? So let's first start and go through the male reproductive system. And when we're talking about the primary sex organ, we're talking about the testes. And these guys are located within the scrotum, so they're outside of the body. So this is really important because the whole job of the scrotum is to protect this developing sperm, so the developing gametes in the testes. So when we talk about the scrotum, we're talking about the skin and the superficial fascia that are surrounding the testes. And essentially, they want to keep the testes at a perfect temperature, which is going to be about 3 degrees cooler than the body temperature. Because if they were in the body temperature, the sperm would die. So any higher temperature kills the sperm, any lower temperature kills the sperm. So it has to be at this perfect temperature all the time, and that's the whole job of the scrotum. And so we have a couple of muscles to help this help the job of the scrotum. So the dartos muscle is the layer of smooth muscle that is just right around the scrotum under the skin. And essentially it's going to cause the wrinkling of the scrotum to essentially help protect the testes and insulate the testes. And then the cremaster muscle is actually a skeletal muscle that comes down from the abdomen, from the body wall, and can elevate the testes into the abdomen or into the body. So essentially, if it gets too cold outside, right, the cremaster muscle is going to elevate the testes into the body so that the sperm does not die. So now let's talk about the testes itself. So we have a couple connective tissue structures and we have some internal structures as well, some tubing. So when we're talking about the external structures or these connective tissue structures, the tunica vaginalis is essentially the sac that encloses the entire testis. And this is really just an extension of the peritoneum. So remember, our peritoneum is that serous membrane. So essentially, the testes started in the abdomen, right, in development, and then they drop into the scrotum. So essentially, that tunica vaginalis is that extension of the abdomen, of the peritoneum. And then there is a tunica that's just right around the testis itself, and that's the tunica albuginea. And essentially it's just a fibrous capsule, and it divides the testis into these lobules. And then each lobule is going to contain this coiled tube work called the seminiferous tubules. So if we look at our picture over here of the testis, let me get my laser pointer out. So the whole testis is going to be enveloped in that tunica vaginalis. So essentially think you have kind of a parietal layer and a visceral layer, just like the peritoneum, right? And then just right up against the testis, and what the testis is kind of made out of is that tunica albuginea. So that's that internal layer. And essentially that's going to divide up into these lobules. Okay, we've talked about lobules kind of in the kidneys as well, so very similar. And within each lobule, there's these this ductwork tubes called the seminiferous tubules. And so those are the yellow kind of squiggly lines. Okay. And then right outside that, kind of connected to those tubules, is the epididymis. So right off and kind of coiled around the testis is this kind of comma-shaped structure called the epididymis. So this whole structure here is the epididymis. Okay. And essentially it's just a continuation of those seminiferous tubules. So we'll talk about the sperm production is going to be in these tubules, then they're going to work their way out into the epididymis. Okay, and we could divide it up into a head region, a body, and a tail of the epididymis. Okay, so we'll talk a little bit more about these structures in a minute. So now the important part about the vasculature for the testis is that it's still all about temperature, right? So when we have arterial blood coming down and into the testis, it's going to be hot, right? It's going to be the temperature of our body. So the arterial blood has to have a way of cooling down before it gets to the testis, or else it's going to kill sperm. So we've talked about the testicular arteries, right, or gonadal arteries already that are coming off that abdominal aorta, so it's the same thing. So that's going to come down through the spermatic cord, which is the structure that contains multiple different structures in it, and we'll talk a little bit more about that. But we do have a testicular artery coming down, but essentially the big difference is we have this interesting vasculature called the pampiniform plexus. And essentially the pampiniform plexus is a venous structure, so part of the veins, so it's deoxygenated blood. And essentially it wraps itself around the artery that's coming in to try to help dissipate some of the heat from the artery. So the pampiniform plexus is the blue structure in the picture. And we can kind of see an artery maybe underneath in red. And so that pampiniform plexus wraps itself around the artery to help dissipate that body heat of the artery. Okay. Now let's talk about those seminiferous tubules. So these guys are the location of sperm production. So in our picture, we've got a cross section of a seminiferous tubule. Okay, so this is one tubule. So we're talking about the lumen of the tubule there. And you can see all of these developing cells along the wall of that tubule. Okay. So if we talk about the different cell types in the seminiferous tubule, we have spermatogenic cells. These are the cells that are going to become sperm. Okay, so they're the developing sperm cells. But then we also have another type of cell called the Sertoli cell. And these guys are just supportive cells. Remember we were talking about the brain and, we have our neurons, but then we have our neuroglial cells, which are the supporting cells. So same thing. These guys, these Sertoli cells are going to help the developing sperm cells. So now if we talk about the process of sperm production, it's called spermatogenesis. So anything with genesis, kind of think, you're producing something new, right? So a spermatogenic cell becomes a sperm cell, but it goes through a couple of stages. So sperm formation, the process begins at puberty, right? That's when all the hormones are activated and all this process starts. It takes about 75 days for, a spermatogenic cell or a stem cell essentially to become a mature sperm cell. So there are these different stages, right? So what happens is the spermatogenic cells, or these more like stem cells, start from the outside of that seminiferous tubule, so right along the wall. And as it develops, it's going to move its way toward the lumen of that tubule. So it's going to be spermatogonia when it's out on this periphery of the tubule. And then it's going to go through a couple different levels. So it'll become a primary spermatocyte, and then a secondary spermatocyte. And then right till they almost get to the lumen, they're considered spermatids. So spermatids, kind of like little sperm. And then once they get to the lumen, they are sperm, okay? So we can talk about spermatogenesis in a couple of stages. So 3 stages. And again, don't get too caught up on, some of the details of these stages of spermatogenesis. So essentially, stage 1 is going to be mitosis. So this is where kind of mitosis and meiosis comes into play, back to biology, right? So mitosis is just the division of one cell into 2 cells. And then meiosis is very specific into reproduction. Okay, so we'll talk about that in a minute. But stage 1, essentially, you're going to undergo mitosis. So you're going to have those spermatogonia divide into 2 primary spermatocytes. Okay? And then during stage 2, you're going to go through 2 levels of meiosis. Okay? So meiosis 1, you're going to get 2 secondary spermatocytes. And then each spermatocyte becomes a spermatid. So you're going to get 4 spermatids total. Okay, so the big take-home message is that one stem cell becomes 4 sperm cells. Okay, because you're going to undergo 3 levels of division. Okay, so if we follow our picture over here, essentially we can follow these stem cells, these spermatogonia up here, all the way down. We go through a primary spermatocyte, then that becomes 2 secondary spermatocytes, and each one of those become 2 spermatids. So then we end up with the 4 spermatids. Okay, so essentially then we have to mature these spermatids. Okay, so don't worry too much about the spermatozoa. Okay, but essentially they have to go through a process of development to mature. Okay, so we won't go through that quite as detailed. So this picture kind of lays out what the division is happening, the cellular division undergoing spermatogenesis, okay? And then with our picture that we went through, what's actually happening within that wall of the seminiferous tubule, okay? So this is the development stage of those spermatids. So they have to mature to become sperm to be able to swim, okay? But here's those divisions that we're talking about. So, mitosis in the beginning, you're just having a division of the cell into 2 identical cells. And then again, we're going to undergo 1 stage of meiosis. So we half the number of chromosomes, and then we're going to divide again and become 4 cells, each with half the amount of chromosomes. Okay. So now let's talk about those supporting cells. So these guys are called Sertoli cells, right? So these guys are kind of wrapping themselves around those developing spermatogenic cells. They're also called cystinocytes, like sustenance, but I like Sertoli cells better. So these guys essentially are large cells. So they extend from the wall of the seminiferous tubule all the way to the lumen. So all this orange material that you see here is all that Sertoli cell. So here's the nucleus of the Sertoli cell. So here's one cell, and you can see that it wraps itself around all of these developing cells. Here's another cell over here. So all the yellow that you see is all the cytoplasm of that Sertoli cell. And essentially there's these tight junctions in between the two cells, so in between the two Sertoli cells, and essentially they form kind of this blood-testis barrier. So they protect the developing sperm cells from the immune system. So we talked a little bit about the blood-brain barrier, so it's very similar in that it tries to keep out anything that's not supposed to be there. Okay. So now let's talk about the hormone control of spermatogenesis. So we talked about these hormones when we were talking about the endocrine system. So one is going to be the follicle stimulating hormone, and the other is the luteinizing hormone. So these are both gonadotropic hormones. So they're produced in the pituitary gland, and they're going to do something with the gonads. So the first one is follicle stimulating. So essentially it stimulates spermatogenesis. So in the female, it's, an actual follicle that's being stimulated, but in the male, it's spermatogenesis. But same idea, right? It's the gamete stimulating hormone, essentially. And then the luteinizing hormone is going to stimulate testosterone secretion by the cells in the testis. So when we looked at our picture of the seminiferous tubules, we talked about, the spermatogenic cells and the Sertoli cells, but just outside or in between the tubules, we have some cells out there that are called Leydig cells. And these guys are the endocrine cells. These guys are going to be the ones that are secreting testosterone. And so the control of the secretion of testosterone is under the control of the luteinizing hormone, which is also produced in that pituitary gland. So now let's talk about the epididymis. We have undergone spermatogenesis in those seminiferous tubules. So we've produced these semi-mature sperm. So they're going to head out of the seminiferous tubules and into the epididymis, which is essentially just one long tube. And it contains pseudostratified columnar epithelium with some stereocilia. And essentially, this is swim school for the sperm. So essentially, it's a 20-day journey for the sperm to move through all that 20 feet of duct work. And this is where the sperm really undergoes the complete maturation process. So essentially, it gains the ability to swim as well as the ability to fertilize an egg through the acrosomal reaction. So we'll talk a little bit more about the acrosome later, but essentially it's kind of a headpiece on the sperm. And it is full of enzymes, so it can break down the wall of the egg. But essentially it fully matures. Think of it that way. So it learns how to swim because it's got to swim through all that ductwork, and it matures along the way. And then in the tail of the epididymis, so over at the end of it, essentially the sperm wait in the tail until an ejaculation. So it's essentially being stored in the epididymis, okay? So it has to get through the long tube work and then it is stored, okay? So when we talk about the sperm anatomy, we said, how does the sperm mature? What does it mean that the sperm is fully mature? So it's matured in the epididymis, and part of that is this acrosomal reaction. So it has to develop this acrosomal vesicle in the head of the sperm. So when we talk about the different pieces of the sperm, it has a head piece, which contains that acrosome, which contains the enzymes to break down the wall or the zona pellucida of the egg, or also called ovum. We'll talk about that. Then there's a mid piece, and this essentially is the motor. So it's full of mitochondria for energy. So then that motor is what whips the tail around, and it's essentially just a flagellum, and it propels the sperm forward. So that's the tail that moves the cell. Okay. So once the sperm has made its way through the epididymis, so now it's a mature sperm cell, it can swim, and we're just waiting for an ejaculation to go through the rest of the ductwork. So first it's going to head out through the ductus deferens, which is also called the vas deferens. And essentially it does do some storage of sperm, but mostly it transports the sperm to the urethra. So it runs from the epididymis to the urethra, and it has kind of a long journey kind of up and around the backside of the bladder. So if we look over here, and it's part of the spermatic cord, so it makes its way from the epididymis up through the spermatic cord into the abdomen, and it curves back around the backside of the bladder. And then this is also the location for a vasectomy. So essentially they ligate the ductus deferens so that the sperm can't finish their journey, right? So if we look at the histology of the ductus deferens, it's essentially very similar to the either urinary system or the GI system. It's got some pseudostratified columnar epithelium, and then it's got some thick muscular layers, right? So it's got to squeeze everything through that ductwork, and then it's got some adventitia on the outside. So very similar to a lot of our other tubules of the body. So we keep talking about this spermatic cord. So there are some important structures that are contained within the spermatic cord. So we have our ductus deferens, right, carrying the sperm. We have the blood vessels, right, our testicular artery coming in, and we have that pempiniform plexus that's wrapping around the artery. We have the muscle, right, our cremaster muscle over here, which wraps itself around the testis and can pull it up into the abdomen. Then we have nerves, right? So we always has some nerves. But the big thing about the spermatic cord is that the superior portion of it is connected to the inguinal canal. And the inguinal canal is the passageway that connects the scrotum into the abdomen. Because remember, we said that it's really just an extension of the peritoneum that's wrapping itself around the testis, right? So we have a connection into the abdomen. So how could that be bad, right? Essentially, there's a hole out into the outside world. But sometimes some small intestine can work its way into that inguinal canal and we get an inguinal hernia. And that can be very painful and even life-threatening to the bowel. So now we've made our way through the ductus deferens and we're going to join with the urethra. So if we look at our journey over here on our picture, we've started in the testis, we've gone through the epididymis, and now we've gone through that ductus deferens all the way into the abdomen, right, through that spermatic cord, into the abdomen to the back side of the bladder. So this is kind of the inferior, kind of posterior, inferior side of the bladder. And we're going to join in with the urethra. And at this junction, it's called the ejaculatory duct because essentially we're going to gain some fluid of the semen from these glands, and we'll talk about that in a minute. And then we're going to work through the different parts of the urethra. So first we go through the prostatic urethra, which runs right through the middle of the prostate gland, and then the intermediate urethra, which is really just at the body wall, okay? And then finally, we're going to go through the spongy urethra to the outside of the body. So now let's talk about the accessory glands. So what's the whole purpose of the accessory glands? So we have 3 different accessory glands, and they're going to produce fluid that's going to make up the majority of the volume of semen. So when we talk about the sperm cells, that's part of the semen, but what is making up the majority of the fluid are these glands. So the first one, or first pair of glands, I should say, are the seminal vesicles. So these guys are on kind of the posterior surface of the bladder. So they sit right here, okay, right next to that ampulla of the ductus deferens, which is just an enlargement of the ductus deferens running into that ejaculatory duct. So this secretes about 60% of the volume of semen, these seminal glands. And what is in this fluid? Well, the most important thing is this fructose, which is sugar. So essentially it's going to nourish the sperm, help give them energy to swim the distance they need to swim. And there's a couple other things in here as well, including some things that enhance sperm motility, prostaglandins, which are hormones that help to stimulate uterine contraction. So essentially to help the sperm move through the female reproductive system. Then there's also some things that help to suppress the immune system against the semen in the female reproductive tract. So the female reproductive immune system tries to kill as many of the sperm as possible because they see them as foreign invaders. So essentially these help to suppress that immune response a little bit. So our second accessory gland is the prostate gland. So this is not a paired gland, it's only one, and the prostatic urethra runs through the middle of it. And this secretes about, 20 to 30% of the volume of semen. It contains also substances to help sperm motility, as well as some enzymes that help to clot the semen, but then also liquefy it. Okay, so to help protection of those sperm for the journey. And then our last pair of glands, again, are the bulbourethral glands. And these guys are very small glands, right kind of at the body wall essentially. So they sit right at that the base of the prostate gland, right on in the body wall essentially. So they are going to produce this kind of mucus secretion called pre-ejaculate. And essentially the mucus is going to enter the spongy urethra prior to the semen. Essentially, it's going to neutralize any acidic urine that came through here, right? Because if you think about it, this spongy urethra is a common tube for both urine and semen. So urine will kill semen, so we want to make sure that we clean out the spongy urethra before the ejaculate comes through. It also helps to lubricate the urethra as well. So now let's talk about the male secondary sex organ or accessory sex organ, which is the penis. So this is not the primary sex organ, which is the testis, but secondary or accessory. So let's talk about the external anatomy first. So we can break it up into a couple different pieces. So we have the body of the penis. the glans penis, which is really just the beginning portion. And then the prepuce covers the glans penis, but only in a non-circumcised penis. So the circumcision removes that prepuce or the foreskin. And then if we look at the internal anatomy, we're really talking about these different erectile bodies. So these are what are going to fill with the venous blood during an erection. So we have three of them. So the first one is the corpus spongiosum, which essentially just wraps around the spongy urethra. So that's why it's called the spongy urethra, because it runs through the corpus spongiosum. And then the two larger structures are the corpora cavernosa. It's hard to say a couple times fast. And these guys are a little bit larger and are on the dorsal aspect, so on this or superior aspect, and the corpus spongiosum is on the inferior aspect. Okay, so we have a paired. That's why you can also call it the corpora cavernosa, which is plural. Okay, or corpus cavernosa. Okay. So now let's talk about the female reproductive system. So the whole point of the female reproductive system is to produce those gametes, but also be able to support a developing fetus. So the internal organs of the female reproductive system are the ovaries. Those are the primary sex organs producing those gametes. We have our uterine tubes, also called fallopian tubes, which essentially carry that gamete or egg into the uterus. So the uterus is where that fetus will develop. And then the vagina connects up to the external environment. So that whole function again is really just to produce gametes as well as support a developing fetus. So the ovaries are our primary sex organ. They're going to be the ones producing our eggs, which we call ova. So ova are the technical anatomical terminology for eggs. And the interesting thing about the ovaries is they're really not attached to the uterus directly, so there is no continuous connection. So we have to have some ligaments and mesenteries to help hold the ovaries in place, essentially. So what are these ligaments and mesenteries? So first we have the broad ligament, which holds not only the ovary, but also the uterus kind of in place. So essentially this whole portion of connective tissue is the broad ligament. And essentially it is a fold of the peritoneum. So it's kind of considered a mesentery. So we talked about the mesenteries in the abdomen. So this is a very similar structure, but just holding and supporting these female reproductive organs. And then we have two ligaments. So we have the suspensory ligament coming into the ovary. Okay, so it's attaching to the pelvic wall, and it's kind of an extension of the broad ligament as well. Okay. And then the ovarian ligament itself attaches the ovary to the uterus, kind of on the medial aspect, so that the ovary doesn't go too far away, because it's kind of suspended here and it's not directly connected other than this ligament to the uterus. And when we talk about the arterial supply, the blood supply to the ovary, it runs through this suspensory ligament to the ovary. So it's bringing in that blood supply to the ovary. So if we look at the internal structures of the ovary, some things are very similar to the testis. So it contains a fibrous capsule, just like the testis, and it's named the same. So the tunica albuginea is the same as the testis. It's just the outer fibrous capsule of the ovary. And then we can split the ovary up into a cortex and a medulla. So we've seen these terminologies before as well in the kidney. So cortex is kind of on the superficial aspect, and then the medulla is on the inside. So the cortex is what is going to house these developing follicles, which contain those developing eggs or oocytes. Oocytes are the ova, right? So same thing. And the follicles really are just these multicellular sacs that are containing those eggs, those oocytes. Okay. And then in the middle, we have these this medulla. So the medulla is essentially just where all the blood vessels, lymphatic vessels, and nerves are all coming in and out of the ovary. Okay. So the crazy thing about the ovaries is that when it ovulates that new developed oocyte, that egg, it essentially ovulates it into the peritoneal cavity, so into the abdomen. So essentially, the whole job of this uterine tube is to try to capture that newly developed oocyte and bring it to the uterus. Okay? And so what are the different parts of the uterine tube? Well, this very first part that kind of has these finger-like projections are called the infundibulum. So the infundibulum is this first portion, and then these little finger-like projections are called fimbriae. And essentially the fimbriae kind of wave themselves around the surface of the ovary, kind of move along the surface of the ovary, trying to pick up any oocyte that may have been ovulated. So then hopefully it picks it up and brings it into the ampulla. So the ampulla is kind of this portion of the curve of the uterine tube, and this is really the usual site of fertilization. So the sperm usually make it all the way up into the uterine tube, and this is the site where fertilization happens. So the egg works its way not too far into the uterine tube before it comes into contact with the sperm. And then the last portion is just called the isthmus, and so it just empties into the uterus. So the uterus is inside the pelvic cavity. So essentially it's anterior to the rectum but posterior to the bladder. So it sits right in between the two. Okay. So what are the different portions of the uterus? So we have a fundus. We've seen fundus before in the stomach. So it's just that rounded superior portion. Okay, so this would be the fundus. Same here would be the fundus. And then we have the cervix, okay, which is essentially the neck of the uterus that opens up into the vagina. And then the cervical canal is essentially the canal that the baby has to make its way through. Okay, so the cervical canal is what is going to have to dilate for the baby to pass through the cervix. Okay. And essentially there's an internal os and an external os. So essentially the opening into that cervical canal. So the internal os is on the uterine side and the external os is on the vaginal side. Okay. So when we talked about the different ligaments and mesenteries of the ovary, but they're very similar and even shared for the uterus. So the mesometrium is part of that broad ligament. So the mesometrium is just the portion that essentially attaches to the uterus, but it's part of the overall large, broad ligament. And that's what attaches to that pelvic wall, holds everything in place. And then we have a round ligament that binds the uterus to the anterior part of the pelvic wall. So essentially it kind of tips anteriorly, so it points a little bit anterior, and it attaches up to that pelvic wall via that round ligament. So the uterus is composed of three layers, right? Just like a lot of other organs in the body. So we have a perimetrium, except everything's with metrium, right? So perimetrium is that outer layer of connective tissue, which is the same as the peritoneum. So it's the same layer, that serous layer of the peritoneum. Remember our serous membrane, so same thing. Then we have a large smooth muscle, which is the myometrium. So this has to contract and push that kid out of the cervix. Okay, so large muscle of the myometrium. And then the endometrium is important because it's the lining of the uterine cavity. And essentially, the developing embryo is going to burrow into the endometrium, and part of that endometrium is going to be the placenta. So it's going to develop into the placenta and support that developing embryo. So now let's talk about some of the female cycles. So we have the menstrual cycle and oogenesis. So oogenesis is the development of the gamete, just like spermatogenesis in the male, right? So we call it oogenesis in the female. And so they coincide, right? So the menstrual cycle coincides with the oogenesis, or the development of the ovum. So the menstrual cycle is a 28-day cycle, right? So it's due to hormonal fluctuations in the ovary and the uterus. So we have two different cycles that are going on simultaneously. Um, one is happening in the ovary and one is happening in the uterus, okay? And overall, we call it the menstrual cycle, okay? So what's happening in the ovary? So the ovary is going to be producing the follicle and the oocyte. Okay, so we'll talk about the ovarian cycle first. And then the uterine cycle is what is happening in the uterus. So the wall of the uterus, or that endometrium, is essentially preparing for the implantation of the embryo. Okay. So we're going to talk about these two different cycles and what is happening. So on the right, we have a lot of graphs and things going on. Don't panic. So essentially, the first table is just showing what's happening with the hormones. Okay. One set of hormones. Okay. So these are our FSH and LH. These are these gonadotropic hormones. So they're going to be stimulating the other hormone production. So then what is happening in the ovarian cycle is this graph. So we'll talk about these different phases, right? So the follicle and oocyte development into ovulation. And then the plasma hormone levels. These are the estrogens and progesterone. So what's happening there? And then the uterine cycle is at the bottom. So what is happening in the endometrium, okay, in the wall of the uterus, okay? So we'll go through these individually, don't worry. So let's go through the ovarian cycle first. So it has three phases. So first we have the follicular phase. So this is the follicle and oocyte development. Then we have ovulation, which essentially once that oocyte reaches maturity, it's going to ovulate. Okay? And that means it's going to be expelled from the follicle and from the ovary itself. And then we have the luteal phase. So the luteal phase is important because it's preparing the uterus for an embryo, for implantation. Okay. So if we look over here on our graphs and tables, we've lined them up. So if we look, here's our ovarian cycle right here. So here's our follicular phase, the development of those follicles and the cells. And then we have ovulation, which is essentially is one event, right? So it's the expulsion of that cell out of the ovary. And then the luteal phase. So what happens with the cells that are left from the follicle is going to produce progesterone. Okay, so it's going to produce this hormone to prepare the body for pregnancy. Okay. So the only point of these guys here, the spikes in these levels, is for ovulation. So you do see a rise in that FSH for the follicular development here. So, but there's a spike in both to signal ovulation. Okay. So the follicular phase, let's go through the development of the follicle and the oocyte. Okay, so very similar to spermatogenesis, but a little more complicated. Okay, so essentially we have these primordial follicles, which you are born with the number of primordial follicles and the eggs inside them that you will have for your entire life. Okay. So the development of these primordial follicles are stimulated by that FSH, that follicle stimulating hormone from the pituitary gland. So one primordial follicle will become a primary follicle. Okay, so it's going to pick one out of the bunch to start developing. So you have kind of this reserve of all these primordial follicles waiting to develop. And then the FSH stimulation is going to stimulate one to develop. Maybe two, and that's how you get twins, but usually one, right? So then that primary follicle becomes a secondary follicle, okay? Just like we were talking about primary spermatocytes, secondary spermatocytes, same thing. So there's some special features that are going to happen in this developing follicle. It's going to develop this corona radiata. So essentially it's just a bunch of cells, okay? And these are granulosa cells. And these guys are going to be what are going to start secreting hormones. Okay. The other important thing that starts during this time is we create this coating around the oocyte, around the cell itself. And essentially it's a glycoprotein coating. I kind of think of it like sugar coating. Okay. Glyco, it's actually a sugar protein coating. And essentially it's very, very strong, and it's for the protection of the oocyte, okay? Because again, you have this developing oocyte, which is the cell, inside this developing follicle, which essentially are mini cells around that developing oocyte, okay? And then that secondary follicle gets bigger and bigger and becomes a Graafian follicle. And the Graafian follicle is what is ready to be ovulated. So it's essentially a mature follicle. It's ready to expel the cell out of the ovary. Okay. So now we have this mature Graafian follicle, this large follicle ready to be ovulated. So ovulation is just a one-time expulsion of that developed oocyte. Okay, so it's about halfway through the menstrual cycle or ovarian cycle, however you want. The menstrual cycle is the overall everything happening. We're talking just specifically about the ovarian cycle. So about halfway through. Okay. And essentially one mature oocyte exits from one ovary, right? So we have two ovaries, so only one undergoes ovulation at a time per cycle, essentially. And so it enters into this peritoneal cavity, and hopefully it's picked up by those fimbria, those little finger-like things, and it enters into the uterine tube. So the signal for ovulation is that spike in the luteinizing hormone in LH, as well as FSH too, but we talk a lot about the LH as being the one to stimulate ovulation. So now once we ovulate or get rid of that oocyte out of the ovary, now we go into the luteal phase. So this is the last half of the cycle. So after ovulation, we essentially leave these follicular cells in the ovary. and they become what's called a corpus luteum. And the corpus luteum, the tissue of it secretes progesterone. And progesterone, think of progesterone like the pregnancy hormone. It's going to prepare for implantation of an embryo. Okay? And if no implantation happens, then the corpus luteum is going to degenerate and the cycle starts all over again. So I like this picture because it kind of puts it all together, right? So it goes through the phases of the ovarian cycle. So the follicular phase, we develop it, then we have ovulation of that mature follicle, and then we have the luteal phase with the corpus luteum secreting that progesterone. And then if we don't have an embryo, we're going to start the process all over again. So when we're talking about the ovarian cycle, we're also talking about oogenesis. So we're talking about follicular development during the follicular phase, but what's happening inside the cell. So it takes many years for oogenesis to be complete. So like I said, we're born with the number of primordial follicles that we're going to have in our lifetime, which is why we talk about women, having a reproductive timeline, because we only are going to have so many primary oocytes. So each month we get the surge of LH, which is going to cause one primary oocyte to complete meiosis, okay? And then we're going to ovulate a secondary oocyte, okay? So we only undergo meiosis II if fertilization occurs. Okay, so it's a little different from the female and the male. So oogenesis is only going to produce one ovum. And then we get these things called polar bodies. You can have anywhere up to 3 polar bodies, and essentially they're not going to contribute at all to the developing embryo. Okay, it's just kind of like extra genetic material thrown out the window. Okay, so it's very different from men, from males. They're going to have, one sperm cell or one stem cell is going to produce 4 sperm cells. And in the female, we only have one stem cell producing one egg or one ovum. Okay. So if we look at what's happening in the cellular division, don't get too caught up in it. It's similar to the male reproductive system or spermatogenesis, but oogenesis only gives you one ovum. Okay, so that's the big take-home picture, okay, is that we get one ovum out of this whole process. Okay, so this is just showing you the difference in what is happening in the cell, the oocyte, and then what is happening in the follicle. Okay, so that's what's it's comparing the two. And again, don't get too caught up in this, but just wanted to show you what's happening. So now let's talk about what's happening in the uterus. So let's talk about the uterine cycle. Okay. So it's really what's happening in the endometrium. So the endometrium is that inner layer of the uterus. So it's in coordination with the ovarian cycle, which they're all under the control of estrogen and progesterone. So what are the 3 phases of the uterine cycle, right? So we have the different phases of the ovarian cycle and they line up. So the menstrual phase lines up with the follicular phase, and what's happening is the endometrium is being shed, right? So there was no embryo implantation, so we shed that endometrium and we get ready for a new potential embryo. Okay, so that's during that follicular phase. We're, taking one primary oocyte and it's developing into a secondary oocyte. And then we have proliferation, right? So we rebuild that endometrium. Once it's shed, then we rebuild it, ready for ovulation. So we said ovulation is about halfway through at day 14. So by day 14, we're going to have a rebuilt, ready endometrium for that developing embryo. And then we have a secretory phase. So we're going to hopefully secrete nutrients for that embryo. So we're hoping that there was implantation of an embryo, fertilization, implantation, and then we're going to secrete those nutrients for the embryo. And if there is no embryo, then the cycle starts all over again with that menstrual phase. So again, now we can look back at all these different tables and see how everything lines up, right? So hopefully it makes a little more sense. So now if we look at our last table here and we look at the uterine cycle, the first phase is the menstrual phase. So we're going to shed that endometrium and then we're going to proliferate or rebuild that endometrium, get it ready right for ovulation, right? So ovulation should happen right before fertilization. And we should have this great endometrium ready for an embryo. If that doesn't happen, then we have that degenerating corpus luteum and lower progesterone, and then we're going to start the cycle all over again. Okay. So now let's talk about some of the secondary organs in the female. So we have the vagina. So that opens to the external environment. So when we talk about the mucosa, we're talking about stratified squamous epithelium. So essentially, we have these transverse folds and stratified squamous epithelium to counteract the outer environment. Okay. Then we also have a layer of smooth muscle called the muscularis and adventitia, which is our fibrous connective tissue on the outside. Now, if we look at the internal structure, we know that the cervix is the opening into the uterus, and on either side of the cervix, we have what's called the fornix. And essentially, it's just these recesses of the vagina. And essentially all these structures, the transverse folds, everything allows for expansion of the vagina, okay, around the cervix to accommodate that baby's head coming through. So now let's talk about the external genitalia for the female. So we have the labia, majora, and minora. Okay, so this is the homologous structure to the male scrotum. Okay, and then within the labia is the vestibule. So essentially it's the space between the labia minora. And it's going to house the opening to the urethra and to the vagina. So if we look at our picture over here, here's our labia minora, labia majora, and then we have the opening to the vagina and the opening to the urethra up into the bladder. And then just anterior to that, we have the clitoris. And the clitoris is the homologous structure to the glans penis. It's the erectile tissue of the female. So if all is successful, we have fertilization. So fertilization is the joining of the two gametes, the sperm and the egg, to produce an embryo. So what happens is the sperm has to undergo the acrosomal reaction, which is going to release enzymes from the head of the sperm to essentially break down the zona pellucida, which is that sugar coating I talked about earlier, that glycoprotein coating to help protect the oocyte. And essentially, a lot of people just think that one sperm binds the wall of the egg and one gets in. Well, essentially, many sperm are going to bind the wall or the zona pellucida, and they're all going to help break down that wall so that one can slip in. Okay? So we get a fusion of the oocyte and the sperm plasma membranes, and then that is going to create a barrier. So what happens is there's a cortical reaction. When one sperm enters, there's some enzymes that are going to create an impenetrable barrier for all the other sperm so that nobody else can get in. Okay, so essentially now we have a combination of the chromosomes from the male and chromosomes from the female, and we get what's called a zygote. So essentially we have half the number of chromosomes in both the egg and the sperm, and now we're going to become now a diploid cell or a zygote. And then we start the mitotic divisions, right? So one cell becomes two, and we're going to get all these mitotic divisions as we work our way to the uterus. And again, when we talked about the uterine tube, that's where the most fertilization happens, is up in the ampulla of the uterine tube. So this is a picture of these sperm cells all attacking this very large egg cell. So the egg cell is the largest cell of all human cells, is the oocyte. It's a very large cell. So hopefully the conceptus, which is the dividing cells, is going to reach the uterus. And now we call it a blastocyst because it's undergone a couple of mitotic divisions. And the blastocyst is what is going to implant into the wall of the uterus, which is the endometrium. And this is crazy because implantation has a high failure rate. It's about 50 to 75%, which is why it takes a couple of cycles to have a successful implantation. So implantation happens about 6 days after conception, so after fertilization. So it takes about 6 days to work its way down the uterine tube into the uterus, pick the best spot on the endometrium, and it implants in the best spot. Because if it doesn't pick the right spot, sometimes it will unimplant and then try to find another spot, and that leads to this failure rate. Okay. Don't worry about really all these different cells and all that's happening in the implantation. I just thought this was a great picture to show you the combining of the blastocyst with the endometrium, which will then become our placenta. So how do we create the placenta? It's a very complex process, so I'm just going to kind of go through what I want you to know. And I want you to know that the placenta is made out of both embryonic and maternal tissues. Okay, so it's half baby, half mom. So essentially there is a structure, or this membrane called the chorion, which is around the fetus. And it has these projection finger-like structures called the chorionic villi. And they're going to project up from the chorion into the endometrium. Okay, so if we look at our picture over here, okay, here's our developing fetus. It's in the amniotic sac or amniotic cavity, okay, and then it's going to have the chorion, which is wrapped all the way around. It's going to send up these little chorionic villi into this endometrium. So here's the endometrium all around this developing fetus, okay? And then here is the placenta, what will develop into the placenta, which is the combination of these chorionic villi and the vasculature in that endometrium, which is mom, right? So essentially what happens is there is going to be nutrient and gas exchange across these chorionic villi. So we don't want the mixture of blood between mom and developing fetus, but it's going to allow for nutrient and oxygen exchange, but also some waste pickup. Okay, so essentially all that chorionic villi is going to act kind of like a fetal lung, right? We talked about the placenta being the fetal lung. So we're going to have nutrient and gas exchange, which is brought in through the umbilical cord into that developing fetus. So nice oxygen and nutrients coming in, wastes going out, okay? And the placenta is formed by about 2 to 3 months it takes for the placenta to fully form. So if we talk about the anatomy of the placenta, we're talking about the umbilical cord connecting to the placenta, right? So the umbilical vein, think of it like pulmonary vein, right? So it's running towards the heart or towards the fetus. So it's bringing in all the nutrients and oxygen from the mom or the placenta to the fetus. And then the umbilical arteries, think like pulmonary arteries, away from the heart, away from the fetus, taking that carbon dioxide and waste from the fetus to the mom. And we have this placental blood barrier. So remember we said we didn't want any sort of exchange between mother's blood and developing fetal blood. So essentially it's going to block any sort of maternal white blood cells attacking any of the fetal blood cells. Okay. So we get this blood pool. So essentially the arteries from mom in the placenta are just going to dump the blood out into this pool around these chorionic villi. And then the villi can take up that blood into the umbilical cord. Okay. Same with the waste coming out, we can dump out the waste into this pool, and the veins of the placenta of mom is going to pick all that waste up and dispose of it. Okay. So essentially we have this maternal blood pool where all this gas exchange and nutrient exchange is happening. So the other part of the female reproductive system are the mammary glands. So the breasts are just modified sweat glands. So that's all mammary glands are. So essentially, if we look at the tissue of the mammary glands, essentially we have these lobules. So again, lobules, they kind of look like grape clusters containing alveoli. Essentially, we've seen all these words before in the lungs. So alveoli, they look like the grape clusters, but these are glands. So these are what are going to be producing the milk, okay? And then the newly produced milk is going to go through these lactiferous ducts into the lactiferous sinus and out the opening or the ducts in the nipple, okay? And all these glands are surrounded by adipose tissue, fat tissue. And essentially, the fat tissue is what is going to determine the size of the breast. Okay, so the mammary glands develop only during pregnancy, right? So that's why a breast is going to get bigger during pregnancy, because not only do you have the fat tissue, but now you have these glands that are going to start proliferating, okay? And milk production actually starts after childbirth. And the stimulation for that is that oxytocin, that hormone, okay? So we're not going to really go through the stages of childbirth, but I have it here just to show you the different stages. But I'm not gonna ask you the different stages on the exam. But parturition essentially is the whole length of pregnancy, and for humans, it's about 266 days or about 40 weeks. And every species is a little bit different, so, roughly 9 months, right? Whereas you talk about elephants, that's 22 months, so it's almost 2 years. If you talk about horses, that's 11 months, almost 1 year. So definitely different for different species. And labor is the event of getting the infant out of the uterus, out of the body, right? So we have 3 stages: dilation, expulsion, and placental stage. So the big thing about childbirth is that you essentially have 2 labors, right? So you have to get the baby out first, and then you have to pass the placenta as well. So you can't forget about that. Because that's, especially in some different species, sometimes if you don't have the full placenta passed, then it can cause problems. So the placenta has to detach from the uterus and be expulsed. So now let's talk about some disorders in the reproductive system. So we do see some cancer in males, specifically testicular cancer and prostate cancer. So testicular cancer is common in those spermatogenic cells, so those early kind of stem cells in the sperm production. But it's cured in about 95% of cases, so it's very curable, which is good. Prostate cancer is slow growing. It is in the prostate gland, which is one of our accessory glands, and it wraps right around the urethra. So commonly you see, painful urination, things like that with prostate cancer. A big risk factor is genetic predisposition. So, if other males in your family have had prostate cancer, it's a high likelihood that you may have it as well. And then we do see some cancers in females too, so ovarian cancer, cervical cancer, and breast cancer. So ovarian cancer is in those germinal epithelium in the ovary, okay? So those follicular cells. And then in cervical cancer, you see it in the epithelium of the cervix. It's slow growing, but we have seen a correlation with the human papillomavirus or HPV, which is a sexually transmitted disease. So there is a vaccine out for HPV now, and it's highly recommended to get it because there is such a big correlation between cervical cancer and HPV. Breast cancer, second most common cause of cancer death in women. So again, we see a huge genetic predisposition in breast cancer. So now you can test for the specific gene that causes breast cancer. So, it happens mostly in women over 50, but not always. So it's always a good idea to get mammograms and keep an eye on it. If you know it's in the family, you get tested and screened more often. But treatment, usually surgical removal of the breast or both breasts, radiation, chemotherapy, as well as administration of hormones. So now let's finish off with the development of the sex organs. So essentially, early on, right, in 5 to 6 weeks, we're sexually indifferent. So you need a Y chromosome to become a male. The default sex is female. So we start with two ducts, and then one is going to start to differentiate and one is going to degenerate. Okay. So remember we talked about those mesonephric ducts in the developing urinary system. It's one of our 3 kidneys we develop. Well, if you are a male, then that mesonephric duct is going to become the male ductwork. And we call it the Wolffian duct. So the mesonephric duct is the same as the Wolffian duct. So this is going to become the epididymis, the ductus deferens, the ejaculatory duct in the male. So if you have that Y chromosome, you will develop the Wolffian duct. And then we have the paramesonephric ducts. Para next to the mesonephric duct. So we have the mesonephric duct and the paramesonephric duct. So we start out with both of these. And then if you don't have the Y chromosome, you're going to develop the paramesonephric ducts, which are also called the Mullerian ducts. So these guys are going to become the uterus, the uterine tubes, and part of the vagina. Okay. So in our little picture, and I think I have a bigger picture here because I know it's kind of hard to see, but essentially we start out with both the mesonephric duct and the paramesonephric duct. Okay. And then by about week 6, you're going to have either the Y chromosome or not. So if we don't have the Y chromosome, we'll develop into a female with the paramesonephric duct. Okay. And then if we have the Y chromosome, you'll develop the mesonephric ducts. Okay. Yeah, so here's the bigger picture, and then it shows kind of the development. So when you talk about the reproductive system through life, there's kind of two big reproductive stages, right? So puberty happens to all of us about between the ages of 10 to 15. So your reproductive organs kind of grow to the adult size, but the big thing are the sex hormones are going to start to be secreted and promote those secondary sexual characteristics. And essentially, the events occur in the same sequence across all individuals, but the age at which the sequences happen, differ between individuals, right? So some people go through puberty earlier than others, and sometimes later. So the age is what varies, but the stages are all very similar. And then if you are a woman, they undergo menopause, right? So um, we get a rate of follicle degeneration. So essentially, we said we only have a certain number of those primary follicles to be able to develop into a secondary follicle. So essentially, at around the age of 46 to 54 in women, ovulation and menstruation stops. So the ovaries stop secreting estrogen, the reproductive organs and breasts begin to atrophy after that age. So after the age about 35, your follicular degeneration increases. So here's just the hormone levels to show you what happens during menopause. So a lot of people, a lot of women take estrogen replacements, so replacement hormone therapy to help counteract a lot of these changes. So here's some learning objectives for our last lecture. Okay, so we made it to the end. You guys have been awesome. I am so sorry about all of this that has happened through the semester with COVID. And I know that wasn't the way we wanted to end the semester, but you guys that have stuck with it, I commend you and I wish you all the best of luck with everything in the future. So we will have our final exam next week. I'll give you all the details in Canvas, but it's going to be essentially like every other exam. I'm not going to put any cumulative material in it because you guys have gone through enough. So anyways, I will see you all Thursday at our review. Good luck with everything.